Hello, welcome to demola, and today's lesson we are covering transport across membranes. How do we get important substances like oxygen, carbon dioxide, chloride and sodium ions? How do we get those from out of the cell into the cell? We're going to cover it in today's lesson. Let's get straight into it. Okay, so first of all, we are going to start with simple diffusion. Now, guys, definitions are so important for this topic. Just memorize these definitions. Cool. The net movement of non-polar molecules from an area of high concentration to an area of low concentration across the plasma or cell membrane. Cool. When we say net, we're talking about overall direction. So I just have to show in this diagram here that you can see here that there's a lot more oxygen molecules on the left compared to the right. So overall, you're going to have movement in this direction towards the side where there's a low concentration. But you can see with the red arrow there, some of them are going in the opposite direction. So it's not all of them are going in the direction from high to low, but most of them, overall, we're moving it in that direction. Cool. I just need to mention as well, when we say high concentration to low concentration, that can be substituted by saying downward concentration gradient. So downward concentration gradient can be a substitute for saying high concentration to low concentration. Now, whenever we are going down a concentration gradient, it's passive, yeah? Doesn't require energy to go down a concentration gradient. And I wanted to show you with simple diffusion, literally it's like this. So you see these oxygen molecules, they're just going in between these phospholipid heads, and they're going through where you have the hydrophobic tails, and they're coming out on the other side of the membrane. So you can see here, it's a small non-polar molecule, fits through the heads, interacts with the hydrophobic region. As you see here, that's why something like oxygen can pass through so easily through the cell surface membrane. Cool. Again, we just need to talk about the factors affecting the rate of diffusion. Surface area, concentration gradient. That's going to relate back to Fick's law, which should have been covered in the gaseous exchange video. Yeah. Remind yourself of that formula. And then we're also going to have temperature, and we're going to have the permeability of the membrane. So just to define that, if you have more cholesterol molecules, you tend to have a less permeable membrane, right? So depending on how much cholesterol we have, it can make your membrane more permeable, less permeable. Just depends, yeah? So just be aware of things like that can affect the rate of diffusion as well. Okay, so here's another one that we might have covered in our GCSEs, but it's osmosis. Now, osmosis, guys, remember, you need to get these definitions down, yeah? The net movement of water from an area of high water potential to an area of low water potential through a selectively permeable membrane. You can substitute that with plasma membrane, cell membrane. I just think for osmosis, it's nice to say selectively permeable, yeah? And water potential, I feel like they don't mention this a lot, and I forgot to say as well, you might see it denoted as that. That's also a symbol for water potential. I'll just circle it. But really, water potential is just the ability of water to move. Yeah, that's essentially what it is. Now, H2O, water, is polar but very small. So it can go through that surface membrane normally. Yeah. Remember, osmosis is essentially just a diffusion of water. So it's pretty much the same thing. It can either come through the phospholipids, through the hydrophobic region onto the other side. Yeah. That's because even though it's polar, it is very small. So you can get away with oxygen just diffusing through, but also there are specifically shaped water channels. As you can see with the yellow highlight, that's what's happening there. It's going through a water channel, and with the orange highlight, it's going through the cell surface membrane. Cool. And also just another thing, we just need to remind ourselves from GCSE as well about hypotonic, isotonic, hypertonic solutions. Yeah. So if we're saying hypotonic, that means the concentration of salt outside the cell is less than the concentration of salt in the cell. In other words, we have a higher concentration of water out of the cell than in the cell. Yeah. So you can see that here. With the O, I will just highlight it just for clarity. See the O here and the I here? Outside the cell, we have a greater concentration of water than in the cell. So again, down the water potential gradient, you're going to see water enter the cell. What's the consequence of that? Hemolysis and a turgid cell. So again, we need to think about the properties of cells. That plant cell has a very thick cell wall made out of cellulose, yeah? So it's not going to burst. The red blood cell doesn't have a cell wall. That will burst. Hence why you can have hemolysis there, and you'd only get a turgid cell here. Now again, you can see the definitions on the screen. I'm not going to read them all out. But for example, isotonic solution is the same, yeah? And you can see equal movement in both directions between outside and in. So you'd have a normal cell if it's a animal cell, for example, and a flaccid cell if it's a plant cell, yeah? And then hypertonic means that we have a greater concentration of salt outside the cell than in the cell. I've forgotten to mark it here, but this would be out and this would be in, yeah? So water's going to move out of the cell, yeah? And you can have crenation where the red blood cell swells up, or it can be plasmalized. Well, because you don't have a lot of water, that means the turgor pressure inside of the cell is going to be quite small. So you don't have that cell membrane pushing up against the cell wall, hence why we have plasmolysis happening. So I hope that makes sense in terms of osmosis. Okay, so we're going to get a bit fun with it now. We're going to introduce something new, and that's going to be facilitated diffusion. Now, let's define it. The net movement of charged or polar molecules from an area of high concentration to an area of low concentration through specific channel or carrier proteins. Now, you see in blue, those are the points. Guys, put it down. That will get you the marks in the exam. Yeah, put them down. Cool. Again, like you can imagine, we're pretty similar to diffusion, yeah? It's just that now we're talking about charged molecules and we're saying specific channel carrier or carrier proteins. I just want to put examples of what this might happen with. Glucose, amino acids, ions. These are the kind of things that you're thinking, okay, this might be going through facilitated diffusion, yeah? Now, we said channel or carrier proteins. There is a difference, but both are intrinsic. Both will span the width of the membrane, both with the ability to open and close, yeah? So we just need to kind of go through this. You can see that in orange is the carrier protein, in yellow is the channel protein. I think I do have the differences put down there, but the main thing that we need to worry about is that this channel protein, yeah, is either open or closed, yeah? If it's open, that sodium is just going to go, a nice one just pop through, yeah? If it's closed, sodium's going to be like, hey, bro, why you close the door, yeah? Sodium will not get through if that's closed. Now, the carrier protein is a bit different. The carrier protein can change its shape. So it might bind to the chloride ion, then change shape to deposit it on the other side. So he's a bit more funky with it, yeah? We are going to get into more detail about that coming up soon. And there we go, soon has arrived. We're going to talk about active transport. Now, guys, definition again, I cannot stress it enough. Definition is so important. So it's the net movement of particles from an area of low concentration. See, we're switching it up. Low concentration to high concentration using specific carrier proteins and energy from hydrolysis of ATP. Guys, I cannot stress it enough. And energy from hydrolysis of ATP. So many people miss out on that mark, yeah? It's going to be another mark in the exam, so just be aware. Now, again, we're going to switch it. If we're talking low to high concentration, we can say against the concentration gradient. So that's the kind of difference that we can have there. Now, stages of active transport rarely comes up, but if it does, you want to be the person that can get those marks, yeah? So first of all, ATP is hydrolyzed, yeah? And when ATP is hydrolyzed, you're going to have ADP, inorganic phosphate, and energy, yeah? They normally maybe won't put water in the formula. I've just done it to show this hydrolysis, yeah? Now, PI and energy is going to be really important. So we're going to have allosteric binding of that inorganic phosphate. Now, allosteric, it just means it's a site that's different to the binding site. So normally, say this wants to transport chloride, for example, there's going to be a specific chlorine binding site, yeah? But the inorganic phosphate isn't going to bind there. So you can see it's not binding at the dip, it's binding somewhere else. And what that's going to do is, first of all, it's going to say, okay, you know what molecule, the one in blue, come on to the binding site, yeah? Just stay there for me, please. Now, we're going to use the inorganic phosphate to change shape with the energy that we release from hydrolysis of ATP. You can see it's changing shape. Apologies for my rubbish diagram, but it's changing shape so that that molecule is coming through the carrier protein and it's going to be released on the other side. So I worked really hard on these diagrams, so I really hope that you can kind of see what's happening here. It's changing shape to then deposit on the other side. So I hope that makes sense. That is kind of your steps to active transport. Okay, so bulk transport. This is the final one we got to talk about. But bulk transport is when we have a large molecule. We have a lot of molecules, yeah? Protein, carbohydrate, for example, if it's a bacteria, a whole cell, yeah? We need to be able to get that either into our cell or we need to get rid of it, yeah? So let's talk endocytosis now. Endocytosis is the process by which we have bulk transport into the cell. And I'm not really too pressed with definitions because the likelihood of them asking you to define this is a bit smaller, yeah? But the main types of endocytosis we have, and for SNAB specifically, I'm not sure if it's too important to know, might be more important for other exam boards, but phagocytosis, you're ingesting solid material. Pinocytosis, you're ingesting liquid material. So you can see here, this red molecule, it's come to the cell. It's like, oh shoot, why are you forming a barrier around me? So the membrane engulfs it like that, and then it's going to form a vesicle. Sometimes you'll see this called a phagosome, especially if it's solid material. So you can see that it's engulfed it, it's now in the cell. Yeah, that's endocytosis. Now, exocytosis, just the opposite, bulk transport out of the cell. So this, maybe it's a phagosome again, could just be excretory vesicle, yeah? It's going to come towards the cell surface membrane. It's going to be like, okay, I'm going to fuse with you. See, the membrane's changed shape around here. And once it's changed shape, sorry about that rubbing out, but it's changed shape, and then the molecule can now get out of there. So, just remember endocytosis as well, yeah? That's exocytosis, endocytosis in a nutshell. just know that endocytosis can either be triggered by direct contact. So say that this is my cell, say that this is my invader or my whatever it is, and that's literally just come onto that. Or there's also potential for there to be a receptor there, and then it binds to the receptor, and then the cell's like, oh shoot, we've got to engulf this guy. Yeah? That is our methods of transport across membrane. Okay, so we've just got 2 little questions here, 2 free markers. Have a go, look at them, do your best, and we will get back to you with the answers. And here are our answers. So, guys, when we know that betulin can't get through the membrane, we need to think about these things, yeah? What are the reasons why something might not get through the membrane? Too large, yeah? So it can't go through those phospholipids, yeah? Maybe again, no channel or carrier proteins is another really good point. Or being polar, yeah? We've got to think, normally if it can get through, it's more non-polar, yeah? So first of all, let's flip it. Let's go to those opposites. And then also let's think, if there isn't a carrier channel protein and it's a polar or a large molecule, it won't be able to get through either. Cool. That's kind of what we're thinking for that part. Now here, I believe the question was, let's just double check. The question was to compare diffusion and active transport. Well, guys, literally, if you think about what each of them is, so diffusion is the movement of your non-polar, your non-charged molecules. It's the movement of those, of high concentration to low concentration, yeah? And active transport, different, is going to be against the concentration gradient. You need to use hydrolysis of ATP, and you need to use a carrier protein. I would remember those immediately, yeah? And you can see here, they've probably said it here. We're doing down the concentration against and against, yeah? We're saying that we said that active transport needs ATP. The flip side of that, so active transport needs ATP. The flip side of that would be diffusion doesn't. We've got 2 marks immediately there. Now, just think about something like this, yeah? It's simple to say something like this. Well, both of them we would have said about going through a cell membrane or a semi-permeable membrane. Fine, yeah? Oh, so it's there as well. Yeah, another one, pass-through protein. So, for example, diffusion, you could argue maybe facilitate diffusion is diffusion, yeah? So you can say about going through intrinsic protein, yeah? I hope that makes sense. But you can see, if you just focus on the definition, then you can get your similarities and differences just from that. Okay, so it wouldn't be an A-star demola video without the tips, yeah? But unfortunately, I don't think there's too many A-star tips that I could think of for this. So like in terms of what you should do, remember the properties of the cell surface membrane. If you need to have a look at phospholipids or the cell surface membrane, that was in the last biology video. Memorize the definitions, guys. Really easy marks to get if you just know the definitions. And practice comparing the different methods. So we had that example of diffusion active transport. I'd say be very comfortable doing that. But if you do stick to the definitions, you should be able to just pick off those points like that. Now, what you shouldn't do, don't make things up. Like, I think this happens a lot in A-levels just in general, but you can't just make things up. Just focus on what you've got. So remember about charged or polar molecules or larger molecules are going to struggle to get through a cell surface membrane. Think about the molecule that you have and just be like, okay, is this going to go through, yay or nay? Yeah. And then I'd also just say, don't confuse the things, like, because this is something that you do at the very start of year 12. It can be really, like, you might just not think it's that important, because in the grand scheme of A-level biology, this isn't that difficult of a topic. So just make sure that you don't confuse models. Like, don't confuse endo and exocytosis. It's easier to do. You might think right now that that's not going to be me, but just be careful. Cool. I'd say that's a wrap for this video. So thank you for listening. I hope it's been very, very helpful. We've got comments below if you have any questions. I'll do my best to answer. Like the video, subscribe. We've also got the TikTok and the Instagram, both at A-star demola. But I will catch you in the next one. Bye for now.